Hello, everyone. Hello, can you hear me? Yes, we're working? Okay, hi, everybody. It's a pleasure to be here today. My name is Jason Vanstone. I am the research analyst with the Antimicrobial Stewardship program with the Regina Qu'Appelle Health Region. I brought my pals with me today. We've got Dr. Casey Phillips and Bob Parker down in the corner here. Casey is our lead pharmacist for the program, and Bob is our program manager. So we contacted Dr. Cameron a while ago and worked out an opportunity to come talk to everyone. We've been spending a lot of time in the dank basement meeting rooms of hospitals talking to people, so it's a pleasure to be here in a nice big open auditorium today talking to some future healthcare workers and other sort of sciencey minded people. So I'm very happy to be here. So what I'm going to do today is sort of run you through a bit of background, just so everyone's on the same page about bacteria and viruses and antibiotics. And then we're going to talk a little more specifically about antimicrobial stewardship, what it is, how it works, what we do as a program, and what it is that we're trying to promote. So I will get right into it. Bacteria. This is really the most important part of understanding what's going on here, is what are bacteria and how do they work, and how this relates to antimicrobial resistance. So I always like to start with this as sort of a big picture. So we've got the tree of life here, sort of a tree of life. Basically, that is the very first living thing that ever came into existence in the universe here. And spreading out from that is everything that evolved from there. So everything that's in red, these make up a group called the eukaryotes. These are every animal that you can think of, anything that runs and jumps and swims and flies and crawls and slithers, anything that's green, all of the plants that you can think of. Anything that is tasty and potentially hallucinogenic, those fungi up there, are algae, the protozoa, which are mostly single-celled organisms that function similar to our cells but live as a single cell. So that's everything that's in that red. And if you're still lost, humans fall down that branch of the tree. The green is a group called archaea. These are, again, mostly single-celled creatures that originally, when they were discovered, were found living in very crazy places, thermal vents and, all kinds of sulfur pools and stuff like that. So they're kind of in between bacteria and eukaryotes. They have sort of bits of both in their life. And the main thing that I want to talk about is the bacteria. So that's everything that's in blue. And as you can see, there are lots of them, and they've been around for a very, very long time, which evolutionarily means they're very, very good at what they do. So, what is a bacteria? This is your basic bacterial cell structure. Basically, everything that happens in a bacteria, everything that a living organism has to do is contained within one cell. And so they have methods of protection. You have to protect the cell from the outside world. So in green, you see the plasma membrane. The yellow is a cell wall. That's something that's very important when we talk about gram-positive and gram-negative bacteria. That's what refers to the presence or absence of this cell wall. And then there's this capsule, and some bacteria have this thick red layer on the outside there, the capsule, for extra protection. Every living thing has to be able to store and share their information. So in a prokaryotic cell like a bacteria, they have this nucleoid where all of their DNA sort of groups into this area in the cell. It's not membrane bound, but that's where all the DNA stays. And then they also have these little red pieces here that are floating around. I'll circle this here. That's a plasmid. So it's an extra chromosomal piece of DNA, and these are very important when it comes to antibiotic resistance and the ability of bacteria to share genes. The other piece here involving growth has to do with these ribosomes, so these little blue spheres you see floating around inside the cell. That's what makes the proteins and basically involves a lot of the processes that the cell uses to grow. Locomotion, so cells got to get around. In this case here we have a flagellum, that big green tail that you see, so that whirls around and allows the cell to move around throughout wherever it's living. And romance. As we all know, there's a romantic side to life, and this is very true in bacteria as well. So in the case that we're looking at here, these green little spikes that are sticking out, these are called pili, pilus for singular, and they are sort of instrumental in the ability of bacteria to exchange DNA, so romance, as we'll call it. So I just want to touch on quickly the difference between bacteria and viruses, because this is something that's very key to the things we're going to talk about. So on the left here, you see a sort of typical animal virus structure. And basically what it is DNA, the viral DNA, inside a protein shell. That's it. That's basically what a virus is. And beside that, there's a bacteriophage structure. So these are viruses that infect bacteria specifically. And there's all kinds of different ones, and they kind of look like these weird space probes here. So they've got their DNA inside this protein shell, and then they have this whole mechanism that's kind of like a hypodermic needle, and it pokes through the bacterial cell wall and through their membrane and injects its DNA inside. And that's pretty much the point of a virus. The viral DNA gets inside whatever host cell it is infecting. It hijacks the host cell's machinery in order to produce the viral proteins that it needs to replicate. And those then assemble viral particles, and they'll burst out of the cell and go on and infect other cells beside them, or they get coughed out by us and then infect our friends and family, because we're gross animals. So there's all kinds of different types of bacteria. They're come in all different shapes and sizes. So there's cocci that are circular or spherical. Some of them like to hang out in pairs. Diplococci, like Streptococcus pneumoniae, one of the big causes of bacterial pneumonia. Anthrax there, Bacillus anthracus, is a bacillus, so it's a rod-shaped bacteria. They'll be found often in chains linking each other. And then we've got these weird bent or spiral-shaped bacteria. And so you can see there's some just light microscope and electron microscope images there of different types of bacteria, all different shapes and sizes. So they're a very colorful group in terms of the different sizes and shapes that they can take. So, bacteria and human health. Bacteria have been around, obviously, way longer than humans, and as long as there have been humans, there have been bacteria to infect us. And this is something that has been a burden throughout human history, and I think is evidenced by a few things, so I just want to talk about a couple of those quickly. Any guesses what Mycobacterium tuberculosis causes? Anyone want to shout it out? Tuberculosis, hey, that one's no points for any house there because that's pretty much a freebie. But yes, this is TB, tuberculosis. We know from skeletal remains that humans have been infected with TB since at least 4,000 years BCE, so upwards of 6,000 years ago. And from genetic studies, we know that it's likely been present in the Americas since about the year 100. And this is kind of what I think is interesting about this is these posters from sort of the early 20th century talking about covering your mouth when you're coughing or sneezing, and don't kiss strangers' babies because you'll make them sick. Seems obvious, but we had to warn people about it. And so these are the types of things where when something makes it into our art and our public health advertisements, to me that's something that shows how big of a burden it is on humans. And we can see that even, this is from 2007, but even as recent as that, there is, throughout sort of that Central America and Sub-Saharan Africa and into Southeast Asia, there is a relatively high, almost 1,000 incidents per 100,000 people of TB still today. So it's something that still affects a lot of people in the world today. Okay, how about Yersinia pestis? Any guesses? Or anyone know? Anyone confident that they know? No? The plague. All right. 10 points for Gryffindor, because that one's harder. Okay, so Yersinia pestis is the bacterium that causes the Black Plague, or the Black Death. And this was something that throughout, during its height in the 14th century, killed 75 to 200 million people are the estimates, which this is about 30 to 60% of Europe's total population during that time and reduced the world population from 450 million to about 350 million. So that's a huge number of people, especially for that time. And again, you can see artwork here depicting people bringing coffins and tossing bodies onto carts. So this was something that had a huge effect on the population in Europe specifically, but as well in other places in the world. And it's still something that here's from 1998, but you can still see there are countries in red here, regions where the plague occurs in animal populations, and in yellow is where there are human cases that have been reported. So it is still something that's around. Luckily, we know how to treat it now. So basically, to sum a lot of really bad stuff up in one slide, prior to the discovery of antibiotics, 90% of children that had contracted bacterial meningitis would die. Strep throat was something that was potentially fatal. Ear infections could spread to the brain and cause severe problems. During childbirth, 1 in 200 women would die from infections that were acquired during childbirth. 3 in 10 people would die from pneumonia. And 1 in 10 people that got a skin infection, so if you had a scrape or a cut or were scratching a bug bite and got a skin infection, there was a 10% chance you would actually lose that limb because there was no other way to treat it. So I think that's just something that's really interesting to think about, considering where we're at today, that these types of things which people normally don't die from were very, very big problems. And as recently as 1910, so just over 100 years ago, 46% of the deaths in the USA were from infectious diseases. So just about half the people that died just 100 years ago was from an infectious disease. Luckily for us, we discovered antibiotics. So a Scottish scientist by the name of, Alexander Fleming, it's written right there, discovered penicillin in 1928. So he was working in his lab. It's kind of an interesting story. He was working in his lab growing plates of bacteria, accidentally left a plate open, went on vacation for a week, came back, and this is what he found. So his plate of bacteria had this fungal colony growing on it, and he noticed this ring around the colony where the bacteria were not growing, and he thought, well, isn't that interesting? That's my Scottish accent. Isn't that interesting? So he looked a little further into it, and what they ended up discovering, let's not go there yet, what they ended up discovering was that the fungus is actually producing an antibiotic that will prevent the growth of those bacteria and actually kills the bacteria. And so penicillium was the fungus, so penicillin is the drug. And so they toiled away in the lab for a number of years, and in 1941 was when penicillin was first used clinically, or our first record anyways, and it was used on a policeman, 43-year-old policeman by the name of Albert Alexander. So Albert was out in his garden pruning his roses one day, got a scratch on the side of his mouth, and that developed into actually a life-threatening infection. He had abscesses that affected his eyes, his face, and his lungs. So luckily for him, he was in the area. They gave him penicillin, and he made a remarkable recovery. And then it's World War II. Not a lot of funding goes into scientific research. They ran out of penicillin, and he died. But the proof of principle was there that antibiotics can work to actually cure people from infectious diseases in the case of bacteria here. So research and development exploded over the next 40 years. So you can see on the timeline there the discovery of multiple different antibiotic groups throughout sort of the 50s, 60s, 70s, up until the 1980s. So what did that mean for us? I talked about the burden of bacterial disease. So this is that stat that I brought up before. In 1910, 46% of deaths were from infectious diseases. So that would be bacteria. That would also include viruses. And by 2010, 100 years later, that is down to 3%. And this is in the USA. So that's, just look at the slope of that line. That's a crazy reduction. And of course, that is due to antibiotics and other vaccines and other advances in healthcare, but certainly antibiotics play a big role in this when it comes to bacteria. The other piece of the puzzle here, or the other piece of information that I find interesting, is the life expectancy. So if you made it to your 5th birthday in about the year 1900, you could expect to live to be about 45 years old. That today is up over 80 years in most sort of Western countries. So that's almost doubling the lifespan just by some of these advances in medicine, which includes antibiotics. So how antibiotics work, and I believe Dr. Kamen has gone through this, but I'll just sort of to reiterate, our bacterial cell, so again our bacterial cell, and there's all these different classes of antibiotics that I talked about. So for example, if you look at the beta-lactams or glycopeptide antibiotics, and there are some examples written up there of what those would be. These are things that inhibit the cell wall synthesis, so that yellow piece there, they will prevent the cell from being able to build its cell wall, and therefore you don't have that protection from the outside world, and the bacteria die. Alternatively, you could affect that plasma membrane, which is cyclic lipopeptides. These are antibiotics that depolarize the plasma membrane. Affecting ribosomes, so aminoglycosides, tetracycline, macrolides, oxazolidinones. This is why I have a pharmacist, because I don't pronounce drug names very well. But these types of antibiotics will inhibit the protein synthesis, so they'll affect the ribosomes in some way or another to prevent the cell from being able to build the building blocks that it needs to live. And of course, you could always affect the DNA, that information storage. So quinolones will inhibit DNA replication. Alternatively, you can use things like sulfonamides. These are bacteriostatic more than bactericidal, so they actually don't kill the bacteria, but they slow them down, prevent them from growing as quickly. And so in this case, this one inhibits growth by inhibiting enzyme activity. So you can see this enzyme in the pathway here that it requires, this guy right here. So these drugs inhibit that enzyme, which slows down that pathway, which slows down the ability of that bacteria to grow. So basically what we're looking at is some of those key areas of cellular life are being broken down or inhibited or stopping them from working. So stopping the cell from being able to protect itself, stopping the cell from being able to grow, or stopping the cell from being able to share its information, to divide and conquer. And here's a key piece there, that this is exactly why antibiotics don't work on viruses, because viruses lack these cellular structures and machinery. The viruses have to hijack the machinery of the cells that they are infecting, and so that is why you can't eliminate a virus with an antibiotic. So antibiotic resistance, that's the next step here. What is antibiotic resistance? And basically what this comes down to is it's a process that comes about through natural selection. So that's Charles Darwin, for those of you who don't recognize the man, the legend. Bacteria, they can create new generations in as little as 20 minutes for some of them. So if you think about that, for a human generation, before we're able to reproduce, we're looking at 12, 13, 14 years. A bacteria can do that in 20 minutes. So with that, every time you reproduce, you copy your DNA. In the case of cells, the bacterial cells, they copy their DNA and then they split one mother cell into two daughter cells. Each time you do that, you have opportunity for mutations to occur, naturally occurring mutations to build up inside the genome. And that can lead to good things for the bacteria if these naturally occurring beneficial mutations could create genes that confer resistance to antibiotics. And it could happen in one of many ways. Changing binding sites, creating proteins. So that's some of the examples here. If we look at the green protein here, if that's a mutation that creates an enzyme that degrades antibiotics, then as your antibiotics come inside the cell, they get chewed up by this guy here, and it just spits out the garbage, and then the antibiotic isn't able to work. You could have changes in membrane structure where you create these efflux proteins, so as the antibiotic comes into the cell, it just gets pushed right back out. Highly beneficial for a bacteria to be able to get rid of things that way. You could also change the binding site of the, where the antibiotic binds to get into the cell. So if you change that binding site, the antibiotic no longer has a site that it recognizes to get inside the cell, and it can't even get in the first place. And then, of course, I talked about the ribosomal issue here, so you can change different sites, different things about the ribosome, which doesn't allow various antibiotics to have an effect there. So this problem of, let me get rid of some stuff here. Now it's going to let me erase, where are we, pointer options, eraser. Okay. So if we, there we go. Bacteria are very promiscuous with their DNA, and that's bad for us because it means they can share things very quickly. So in order for us to share a new mutation, something that is beneficial, we have to have our children, and then the genes get passed on, and then we wait 12, 13, 14, 20, 30 years, and they have children, and then it gets passed on, and eventually we have superhumans, but not yet. It takes a long time. Bacteria, it doesn't matter as much for them. They can replicate really quickly, and they have all these neat ways of sharing information. So for example, bacterial transformation, this first one here. Bacteria are able to actually uptake DNA from the environment. So if you have a cell that's died and it breaks apart and the DNA sort of disperses in the environment, other bacteria can take up that DNA, and you can see there if it's an antibiotic-resistant gene, that's something that's very beneficial for a bacteria that was not resistant before and now is. And it did not have to evolve that process on its own. It just takes up the DNA from something else. I spoke of bacteriophages, so these bacteria that specifically infect, viruses that specifically infect bacteria. And so this is called bacterial transduction. So when you get the viral DNA integrates into the bacterial genome, and then as that gets cut out to reassemble new viruses, sometimes little pieces of the bacterial DNA can get taken with it. And those viruses go on to infect another cell, and when they inject their DNA, now all of a sudden you've got a antibiotic resistance gene that has been transferred. And then back to romance. Everyone likes to talk about sex, so bacteria do it too. And that's where this pilus comes into place. So I talked about these plasmids, these circular pieces of DNA. They can carry resistance genes on them. And those can be shared from cell to cell by connecting these two pili. And the plasmid just shoots right through that. And you have now been able to share with something a gene that it did not have to evolve on its own. So that makes for big problems for us in terms of combating antibiotic resistance, because these bacteria can share these genes very easily and very quickly amongst members of their own species and even other closely related species. So what does that mean for humans? This is a human, in case you don't recognize that. Hopefully you do, because you're all going into nursing, or most of you. And we all have bacteria in our guts, all over our body, in fact, but there's a huge reservoir in our guts. And most of these are good bacteria. They're there to help us digest. They make us fart, which is hilarious, and all kinds of other good things that they're there for. And by chance, you could get either ingesting in food or from one of your friends, a antibiotic-resistant bacteria, or perhaps it evolved on its own, just because you're unlucky, inside your gut. And those bacteria could even share with a few of their friends living around inside your gut. But the point here is that for the most part, they're kept in check by all the good bacteria that are in your gut. It's harder for them to grow because they're producing these extra proteins that are not necessarily needed. And so all the other beneficial bacteria can grow a little bit faster and sort of outcompete. The problem is if you get sick and you have to take an antibiotic. Or worse, if you're not actually sick with a bacterial infection, you take an antibiotic anyways, and we'll talk about that. But you take your antibiotic, goes into your system, kind of goes everywhere. And I heard a talk on the weekend, and it kind of struck with me, is antibiotics are really dumb drugs. They don't know where the infection is. They just kind of go everywhere and try and kill whatever's there. So they go there, they kill all of the bacteria that are not resistant to that antibiotic. And that's fine, it'll get rid of your infection, hopefully, but when those antibiotics go away, what's left is the resistant bacteria. And they no longer have to compete with all of the good guys that were around, and now you have a problem. Now you have potentially a resistant infection or something that has now grown in strength and could affect you at another time. So this really emphasizes why it's important to take antibiotics only when they're necessary. And that's not to mention other potential side effects of these medications apart from antibiotic resistance. And those we'll talk about too. So, antibiotic resistance is a process that occurs with every new class or specific antibiotic that we create. We know this. It happens. There's a graph to show it. On the left is when a new antibiotic was introduced, and on the right is the amount of time before we started to see resistance in certain species. So you can see some of them are maybe a decade or two. Some of them, like levofloxacin there, as soon as we started using it, we knew there were bugs that were resistant to it. So it's going to happen with every new antibiotic that we have. It's just a natural process. It's evolution. It's what happens. And the problem is that we are seeing these skyrocketing rates of antibiotic-resistant infections. So methicillin-resistant Staphylococcus aureus, that's the blue line there. You can see since the 1980s, and again, this is American data, but the trend is the same. We get these skyrocketing rates where things are going up and up and up in terms of the amount of resistant infections that we see. And this process, this antibiotic resistance, is driven, for the most part, by the use, the overuse, the improper use of antibiotics. The more we use them, the more resistance we see, the harder it is to treat. We have to try other antibiotics, resistance grows to those, and the problem continues. And again, there's evidence to support this. This here is a graph looking at basically the lag time between resistance in E. coli to ampicillin and our use of ampicillin throughout the year. So along the bottom here we see in January, February, March, April, we can see our resistance here, our use of antibiotics dropping. So that's, right at the peak of cold and flu season. There's a lot of people going to see their doctors, oh, I'm sick, I need something, and they get an antibiotic. And then in June, July, August, in the summer months, that goes away, flu season's over, everyone's feeling good. And then we get back into the winter and people are bugging their doctors again, and they're getting more antibiotics. And what's the point of this is that in the orange line here, which I'm going to go over in red, we see this also sort of peak and trough of resistance to ampicillin in E. coli. So as we start using less antibiotics, going down here, we see less resistance, and it's got about a month lag time there. And then as we ramp up our use again, we see more resistance. So we know that this happens. The more we use antibiotics, the more resistance we see, and the less we use, the less we see, and that's kind of a key point here. So another problem for antimicrobial resistance and our ability to combat this is a lack of new antibiotics. So this is the first chunk of that timeline that I showed before. So that was that boom in antibiotic production. We were finding all kinds of new things on our desks, in our bathrooms, on our telephones, our rotary phones, and picking all kinds of things off from other bacteria, other fungi that were floating around, and that were having their own little war amongst each other, and we just kind of took those and used them for ourselves. And then we got to the 1980s, and everything just kind of stopped. And I guess really the big question is why? So we've had a few since then, but for the most part, there really have been no new classes of antibiotics that have been created in a long time. And that's a big problem for us when it comes to antimicrobial resistance. So I just want to quickly touch on why. One of the pieces of that is the rigor of the drug development process. So for a drug company to create a new drug, from finding a chemical to actually having something that can be used in people, I'll just go over this quickly. Basically, if you look at 10 to 15,000 new compounds that they usually do in their initial searches, 10 to 15,000, by 3 to 6 years of research and development, you might be lucky to get that down to 5 compounds that you want to continue on for, that are considered safe and continue on for testing, so they're not, growing new limbs in people or killing them outright. And so we move into these clinical trials, and they go through phase 1, phase 2, phase 3, where we increase the number of people who, first of all, are willing to take it and then might actually benefit from it at some point. And that could be about a 6 to 7 year process. And from that, you might be lucky to get one drug that is FDA approved. It has to go through some post-approval processes, but that might actually make it to market. So what we're looking at is a 10 to 15 year process that will cost a drug company about $1 billion or more just to get one drug to market. So over $1 billion. And that sort of compounds on a number of other issues. So if I'm a drug company, and I just picked these 3 off of Google Images, not to pick on them specifically, but so there's the cost and time to develop a new drug. One of the other problems is what we sometimes call a new antibiotic introductory lag time. So when we have finally come up with a new antibiotic, look at this one, it's cool, it's going to kill stuff, but let's not use it. We put that on the shelf because we have antibiotics that are mostly working right now, and we want to wait until those antibiotics stop working before we start digging into our back pocket to pick out the things that are still working. And so from a business perspective, that doesn't really work well for drug companies, because this thing that they spent $1 billion in 15 years creating now sits on a shelf for 5, 10, 15 more years, and they don't get to make any money off of selling that drug. The other piece that ties in there is the time limit on patents and competition with generics. So when a drug company creates a new drug, they have a patent, they get to sell that drug exclusively. But again, if we've put that drug on a shelf for 10 years, that patent runs out, and by the time we start using that drug, other drug companies can make their version of it, generics, and it drives the price down. So the initial company doesn't get to make as much money. So, you know, it's a sad but I think true fact that these companies are businesses and they're in it to make money, and it's just not good business for them to make drugs, to make antibiotic drugs. The other piece is no more low-hanging fruit. So as I said, we kind of went through and found everything that was easy to find in terms of fungi and other bacteria that are around us, and trying, to find antibiotics from those. We're at the point now where we're sending scientists into the canopy and jungles to collect sloths who have fungi that live only on the backs of sloths in the Amazonian canopy, to try and find antibiotics from those guys. Or we're sending them deep into caves that no one's ever looked in to scratch bacteria off the wall to try and find chemicals that they use to compete with each other, which is very cool but very difficult work. And you know, your rate of finding new drugs has definitely decreased in terms of the speed that it's happening. So the reality is there's just not a lot of money in it for a drug company, which is something that we'll probably need to address if we want to encourage them to create new antibiotics. So what does this all mean? The problem for us: increased rates of antibiotic-resistant organisms and decreased rates of new antibiotic discovery are leading us to a point where we will no longer have antibiotics to treat infections. And this has never been more true than just this last spring, where there was this posted thing in the Washington Post, a paper that came with it. There was a patient in the U.S. who had a E. coli infection that was harboring this MCR1 gene, which has resistance to some of our last line antibiotics. So antibiotics that are normally not used because they're very hard on us as well. They can cause kidney problems and all kinds of stuff. So you might cure your infection, but you might also mess up your kidneys. So this is something now that if this gene, not if, but when or as this gene continues to spread, if it makes its way into bacteria that are already multi-drug resistant, they're resistant to just about everything we have except these last-line antibiotics, then we actually have truly a first pan-resistant bug. There's nothing that we would be able to do to cure someone if they got an infection with this type of bug. And that's kind of a sobering thought and kind of scary. And the other problem here is that not only do we need our antibiotics to treat an active infection, so if you get sick, you can go to the doctor, you can get an antibiotic, but we also require them for prophylactic or preventative use in many medical specialties. So oncology, people get a huge whack to their immune system when they're undergoing cancer treatment, and antibiotics are very key in preventing sort of opportunistic infections and things like that. So if all of a sudden you're getting a bug that's resistant to things, your cancer may not kill you, but an infection might. Surgery, a lot of hip and knee replacements, especially in an aging population, made very safe by the ability to use antibiotics to prevent infections and to clean up anything in a surgical site infection that does happen. Again, if we're looking into a post-antibiotic era, that might not be so easy. You can choose to either have a messed up knee for the rest of your life or potentially have a life-threatening infection. Obstetrics, again, C-sections, things like that are made much safer by the use of antibiotics to prevent infections. So we're not going back to a point where 1 in 200 women die from infections in childbirth. Another problem in the veterinary world, So by weight, and I put a little star there, but by weight, more than 70% of antibiotics are used in the food animal industry in order to promote faster growth and to basically compensate for the unsanitary conditions in which these animals are raised, which increases production and profit for food producers. And I put the vets get really offended by this slide, and somewhat rightfully so. This is, when you consider the weight of a cow versus the weight of most people, you're not really comparing apples to apples in terms of comparing the weight of drugs that we use. You certainly have to use more antibiotics in a large animal like a cow, or when you have a barn with, 100,000 chickens in it. But the point here is that they're still being used way too much in animals as we are using them too much in humans. And again, the point is we need antibiotics to treat infections in animals, whether that's our pets or our food, just as we need them to treat people. And the inappropriate use of antibiotics in animals drives resistance just as it does in humans. Okay, so this, I think, just tells a good picture of sort of things moving through the environment. So this is us. We're people in our communities living our daily lives. And this is George, and George gets a tummy bug, and George has to go to the hospital. And he gets antibiotics. And unfortunately, through some method, George develops resistant bacteria in his gut. George goes back home with his bugs in his gut. George isn't particularly good about washing his hands, and he goes to the grocery store, and he gets his poop on the bananas, and you buy bananas because they're yummy, and you take them home, and you eat them. Well, now you can have some super resistant bacteria living inside you from a banana or an apple, whatever your favorite fruit is. Perhaps, separately, George goes to a hospital because he's really sick, or he goes to a nursing home or another inpatient care facility. His resistant bacteria spreads to other patients on surfaces. Again, he's still not good at washing his hands, so he's walking around holding the railings, sliding his poop along the railings. Another patient comes in, and it's just a mess. Those patients go home, same thing happens. Potentially, healthcare providers, who I think Dr. Cameron touched on, are notoriously terrible at washing their hands. I don't know why. I think the stat I heard on this weekend was in Canada, we are at a 60% rate of hand washing, and that's one of the better ones. So around the world, we need to do better at this. I'm going to harp on that as we go along. So healthcare workers spreading it around. In the animal world, animals, they get sick, or they're not sick, but they're given antibiotics anyways. They can develop bacteria, antibiotic-resistant bacteria in their guts. That becomes part of the meat, gets into the food chain. If you don't cook your food properly or you're mishandling things, you can get those inside of you. Animal poop goes everywhere too. It's used as fertilizer. It gets into the water, and it spreads those bacteria around. And it all comes back. So there's these big cycles of bacteria and poop and things going everywhere, and it's, we have to find a way to do something to stop the bad ones from becoming a part of this. And if we maintain the status quo, this was a projection done by a group in the United Kingdom, that by 2050, more than 10 million people will die each year from antibiotic-resistant infections. And to kind of put that into perspective, you can raise your hand if you know someone who has been affected by cancer. I'm sure pretty much, yes. And raise your hand if you know someone who's had an antibiotic-resistant infection. So quite a few less people. The point is that will switch. We will all of a sudden start seeing we're basically going to go back to 1910, where bacteria are going to be the #1 killer. These antibiotic-resistant bugs will take over cancer. And that's not some place that we want to be. So what can we do? As I said, when things get to be a real problem, they start to make their way into our culture in the form of artwork. This one's my favorite down here. Did the hospital staff do this to you? No, the hospital staff with a PH. That's hilarious. Because they don't wash their hands. So let's talk about what we can do about it. It's the Antimicrobial Stewardship Program. So we're a new program. We started in June. Our mandate is to try and spread the knowledge and teach people about this and monitor what we're doing in the hospital, in the community, so that we can make active strides to fixing this problem and combating antibiotic resistance. So the question comes, what is antimicrobial stewardship? It's basically a healthcare initiative. So the goal of any healthcare initiative is to promote optimal patient outcomes, and that's our goal. We want what is best for the patients. And so when we're talking about antimicrobial stewardship, whenever an antibiotic is being used or being considered for use, we need to ask ourselves 5 questions. And the first one is the antibiotic necessary? And that's really a big question because we can stop a lot of unnecessary use by not prescribing an antibiotic when we have a viral infection. And if we do need an antibiotic, that's fine, good. We're supposed to use them when we need them. But we want to make sure we're using the right drug at the right dose for the right duration, so the right length of time, and in the right route. So whether that's IV or oral. And I'll talk about all of these here as we go through. So this man is coughing or perhaps choking on his weird scarf there. But basically, you get a lot of people who will come into a clinic. They got a nagging cough. Doctor, I've been coughing for 2 weeks or maybe 3 days because they're wimps, and I just can't get rid of it. It's got to be pneumonia. I had an antibiotic last time. Can we just do something about it? And for the most part, we know that these are bacterial illnesses. Most upper respiratory tract infections are viral illnesses. And so an antibiotic is not going to do anything about that. But that's what we have to ask. Is this actually pneumonia, as the patient might think? Or is it the lingering symptoms of a cold virus? Another big one in the healthcare system is elderly patients who become disoriented in a long-term care home. They're not acting like themselves. Usually the go-to is, oh, they've got a urinary tract infection. Let's give them some antibiotics. They'll be good to go. And again, we have to question, is this actually a urinary tract infection? Or perhaps they're dehydrated, and that's what's causing them to be a little loopy. So really questioning, is an antibiotic necessary? And looking for the signs and symptoms of bacterial infection, things like an elevated temperature or white blood cell count, checking vital signs, respiration, heart rate. And a lot of these things can cross over with viral infections, but certainly there is, it helps to make an informed decision. When you look at radiology data, if you're considering pneumonia and you look at a chest x-ray, and of course laboratory data is great too, to see if you actually have a bug and what bug that is that you're trying to fight. Is it the right drug? So a lot of times in healthcare we do an initial diagnosis based in an empiric therapy, so where you're making an initial treatment of an infection based on the best and most recent evidence that you have. So this might be guidelines, national guidelines for how to best treat pneumonia, again, as an example. Medical studies, things that have been done recently, oh, we've showed this drug works better than this drug. Could be a local algorithm, so in the RQHR, we have algorithms for various different types of illnesses and things like that that our physicians and other healthcare workers can follow. And a local antibiogram. So who's heard of the term antibiogram? Anyone? Okay, obviously the professor. So an antibiogram is kind of like a roadmap for what bugs in your local region are resistant to which drugs. And so we prepare one. I shouldn't take credit for it. Our microbiology lab prepares them. And basically, this is kind of what you're looking at. So if we were considering pneumonia as something that is affecting our patient, then probably, Streptococcus pneumoniae might be our bug that we're thinking at. And so in the region, in the last year that the antibiogram was done, we had 148 isolates of this, and of those, we show across here which ones were resistant and which drugs those isolates were resistant to and which ones they were susceptible to. So the green are the drugs you might consider using because they have the most effect on the bacteria, and then the red are the ones that they're resistant to. So we probably wouldn't choose erythromycin because 64% of the isolates were resistant to that or were susceptible to that. Yes. And so those are based on things like this here. So we put the bacteria on a plate and they drop these little discs on that have drugs, antibiotics in it, and those antibiotics spread out into the plate. And these are resistant bacteria and these ones are susceptible. So it's that easy. And it gets put into a little chart so that our healthcare workers can, when they're doing empiric therapy, have something that they can go on. And I just realized I drew all over my screen again, which I'm going to need. Go away, go away. Okay. So the next step then, you've got your empiric therapy. This patient is clearly sick. We need to start them on something. Let's do this. Okay, they get their drug that we think is going to be the best option. The other piece of this puzzle then is targeted therapy. So when we're thinking about the right drug, We have our patient, we take blood tests or something like that, the radiology tests, and we then are able to make a more informed decision based on those test results. So maybe it wasn't strep pneumonia, maybe it was a different bug, and it'll have a different susceptibility profile, and so we might decide to change our antibiotic after we get those results back. So this is more targeting that therapy to something specific. So then the question of the right dose. And this could depend on a number of things, the type or severity of infection. So some infections will need a higher dose. For example, if you have an upper urinary tract infection versus a lower urinary tract infection, that'll change the type of dose that you'll need. The site of infection, if you have hard-to-hit areas, if you have an infection in the central nervous system where the drugs have to be able to cross the blood-brain barrier, if you have a bone infection, something really deep inside the body, heart valves, which are hard to get antibiotics to, that might change the dose that you need to increase the concentration of the drug at the active site. And of course, there are patient-specific factors, so liver and kidney function will affect, if you have a patient with lower renal function, they'll process the drug differently, and so you might have to adjust your dose based on that. The right duration. So different infections need to be treated for a different length of time. So for example, an uncomplicated urinary tract infection, you might only require 3 days of therapy. Doing 7 days is probably overkill. You're exposing a patient to more antibiotic than is necessary. A cellulitis, a skin infection, maybe 5 to 7 days. If it's diabetes related, maybe 14 days. If it's in a bone, maybe up to 3 months of antibiotic therapy. It can go that long. A prosthetic joint infection, 6 weeks perhaps. Endocarditis, an infection in the heart, 2 to 8 weeks, depending on the type of bug or the valve that's involved. So there's a lot of different things to consider. But one thing we always want to remember is that the prolonged use of antibiotics drives resistance. So we always want to use the appropriate length of time to treat our patient to make them better from what they're suffering from and not give them something else like a resistant infection. The right route, this is something we traditionally struggle with in our hospital. So patients come in, they're sick, they get an IV antibiotic, and they tend to stay on that until they're let out of the hospital. And we want to try and switch people to oral drugs if they're a better option. The less time you have an IV in you, the less chance you're going to have of picking up another infection from that. And other things that can happen. And so this will depend on a number of factors, the type of infection, the type of drug that you're using. If you have a drug that doesn't necessarily have a good bioavailability as an oral agent, then you might be stuck with IV, but things to consider. Also, patient factors, are they clinically stable? Are they on other oral medications? A regular diet, do they have a functioning GI tract? So this is one of the problems we see in the hospitals. People come in, they get on an IV, and then they're taking other pills and they're eating their food, and it's like, well, why can't they be on an oral antibiotic? Let's do that, and it'll be better for our patients. So, who does antimicrobial stewardship? Who does this? And my short answer is everyone, really. This is a job for everyone in our society to play their part. And we try to promote the idea that antimicrobial stewardship is really a collaborative effort among all healthcare workers. So speaking specifically amongst that sector. It doesn't matter if you're a physician, if you're a nurse, if you're a pharmacist, if you're a dentist, optometrist, other people who can prescribe antibiotics, veterinarians, if your patient has 2 legs or 4 legs, we all have to be doing a better job to use our antibiotics properly. And where does antimicrobial stewardship occur? So again, my short answer here is everywhere. It doesn't matter if you're in a hospital, if you're in a long-term care home, community clinics, pharmacies, even in your own home, making sure you're taking your full length of your medication and you're not, if you have an antibiotic prescription, you don't stop once you start feeling better, because that will allow the opportunity for any antimicrobial resistant bugs to take off. Urban, rural, this isn't a problem of big centers, countries with a large population. It's not a problem in India, it's not a problem in Chicago, it's not a problem in Toronto. It happens in Regina. We have antibiotic resistant infections. It happens in Yorkton. It happens in Swift Current in Saskatoon. I see you guys. So there's, this is everywhere, and it's something that we need to combat. So that's a great idea. We should do antimicrobial stewardship. But how do we actually do it? What are the on the ground and the front lines, what are the steps we can do? Which is really, what do we, as an antimicrobial stewardship program, do? And one of the big pieces is called prospective audit and feedback. And basically, it's us going out into the hospital or into community clinics and checking what's happening. Are patients being treated with the right drug, the right dose? Are the ones who are being treated, are those antibiotics even necessary? And so sometimes we'll look at maybe specific antimicrobials. Are we using too much vancomycin? It might be a specific disease state. One of the things that we're looking at is urine cultures and treatment of asymptomatic bacteria. So when people have bacteria in their urine but don't actually have an infection, they don't have any symptoms of a urinary tract infection. Formulary restriction and pre-authorization. So this is another big piece in the hospitals, really, where certain drugs are sort of kept in a separate list so that not everyone has access to prescribing those. It might only be the infectious disease physicians who are able to prescribe those drugs, so you need a consult in order to get them. And those would be particularly the big guns, as we call them, so those broad-spectrum antibiotics, some of those last resort antibiotics. Because really, if someone comes in with something that you can use a specific antibiotic for that will affect just that bug or a smaller spectrum of bugs, we don't need to be trying to kill mosquitoes with cannons. It's just overkill, and it's kind of a waste of resources for those large, broad antibiotics. There are certainly other things that we can do, so as I've called them here, supplements to antimicrobial stewardship. So we have guidelines and clinical pathways, as I talked about, just things that give people information about what are the best ways to treat certain infections. Antimicrobial order forms, streamlining and de-escalation of therapy, so that's getting people on shorter duration or going from those broad to narrow spectrum antibiotics. Dose optimization, IV to oral, all of those pieces that we work at. Of course, education is a big piece. That's why we're here today talking to all you lovely folks. So we've talked a lot with healthcare workers within the region, different groups, so current healthcare workers, future healthcare workers, telling why antibiotic resistance is a problem in medicine, trying to educate people on what the best practice is for treatment of various diseases. And another big piece is how, for healthcare workers, how can you help to educate patients and families about the problem of antibiotic resistance? Because that's really a big piece too, is we need the general public to be more aware of this so that people of all ages, and by extension our governments, fully understand the severity of the problem of antibiotic resistance and the importance of antimicrobial stewardship. Another piece that we do is research. That's my background, that's my passion, so that's something that we've tried to build into our program. So for the most part in Canada, most antimicrobial stewardship programs, if they exist, are relatively recent activities for healthcare facilities. It's something that has recently been mandated as a required organizational practice by Accreditation Canada, which gives us our big stamp to say you're doing well. And so for us, one of the important things is we need to contribute to this research and contribute to the knowledge about how antimicrobial stewardship programs work, what works, what kind of things can we do, what kind of initiatives can we have. And so that will allow us to better understand the ways to disseminate this information and to really create this culture change that we require in medical practice regarding antimicrobial use. So, as an example, over the summer, our program performed a survey of community healthcare workers to try and better understand their knowledge and attitudes towards antimicrobial stewardship. And one of the things we got from them was education is a big piece. Healthcare workers need to know more about it. The public needs to know more about it. We need people to understand what this problem is. And so that's been something that we've been trying to concentrate on a lot this fall. So, what can I do? What can I do? I'm just one person, maybe I'm a future nurse. What can I do? So as a member of the public or as a patient, wash your hands. Just wash your hands. That is the best way to prevent the spread of infection, really. If, as a patient, you can ask your healthcare provider if antibiotics are necessary. A lot of physicians, research shows, have this sort of predisposition in their head that patients are coming in with the expectation, if they have a cold, that they want an antibiotic. And that's not always the case, or at least in a survey that was done, a lot of patients said, I'm not expecting an antibiotic, but it would be nice to have a care plan or something like that, so I'm not just being sent away, but tell me what I can do to help treat my symptoms. And so that's a very important piece is, you can ask if your physician is going to prescribe you an antibiotic, you can ask if that's necessary. And certainly not requesting them if your physician has said they're not. There is definitely some of that goes on. Of course, speaking with your friends and family about the dangers and misuse of antibiotics, you can talk about this awesome lecture that you guys saw today and how much you learned. And, you know, we're going to go talk to our friends and family and tell them the exact same things, because it's important for everyone to know. As a healthcare worker or a future healthcare worker, again, wash your hands often, please. It is the most important thing you can do to not spread things to your patients and spread diseases throughout the hospital. Just wash your hands. We could all pledge right now. We'll put our right hand up. We'll all pledge that we're going to wash our hands at every up. There it is. Okay, thank you. I'll take one. That's good enough. And then certainly, you know, as you're working, wherever you end up working, following the antimicrobial stewardship guidelines specific to your area of what you're doing when caring for your patients with either potential or active infections, so that we're ensuring that our patients are getting the right drug dose, duration, and the right route of therapy. One of the things that we always like to touch on, because we heard this in one of our earlier presentations amongst some nurses that we were talking to, was the question of, I'm just a nurse. I'm just here. I do what the doctor tells me. And that's frustrating, and it's so wrong, because what we're trying to create is this culture of a healthcare team where, you know, the doctor's word is not the only thing that matters. As nurses, you are really the frontline care. You're educators for, you know, patient education. You're leaders, mentors, especially as you grow and continue in your career, and certainly a patient advocate. So many nurses know the patients that they're caring for better than the physician, who might only be coming around the ward, you know, once a day kind of thing. So if you see a patient on an antibiotic and you don't think, for example, they've potentially thought it's a urinary tract infection, but they don't have any symptoms of a urinary tract infection, bring up the question, should this person be on this antibiotic? And that's a very important piece of this puzzle, because as I said, this is a collaborative effort amongst all healthcare workers, and including our patients and their families. So please remember that as you go forward. So I think the last point that I have here is that we really need to change the way that we as a society view antibiotics, and trying to think of antibiotics as a societal drug. So these are things that belong to everybody. Everyone needs them. Everyone's going to get sick at some point in their life and require antibiotics. And it would be nice to know that if it's you, the drugs are available for you to use to make you better. And antibiotics are also a limited resource. So as I've talked about, every time we have a new antibiotic, it's only a matter of time until we see resistance. And so inherent in that is that every new antibiotic has a limited number of uses before we begin to see resistance. So the more we use it, every time we use it, that's one less use that we get as a society for someone next to you. And so particularly thinking about if you have a cold and you just want that antibiotic because you think it's going to make you better, you're using an antibiotic, meaning that someone who's really sick with a bacterial infection the next time around might not have that antibiotic to use because now we've got resistant bacteria. That's what that is. Another thing is this sort of kind of, oh, it's just an antibiotic. We'll just put them on an antibiotic. It's no big deal, you know. And it is a big deal, because antibiotics are drugs, and like any drugs, they have side effects. You know, the most common ones are sort of the annoying ones, the vomiting, diarrhea, nausea, headaches, things like that. But they can certainly have, you can see down under ciprofloxacin there, heart attacks, joint problems, hallucinations, gastrointestinal bleeding. So they can actually have serious side effects. So again, if you think about a patient who has a viral infection and gets an antibiotic, we know the antibiotic is not going to fix the viral infection. And in fact, the only thing you're doing is exposing that patient to these potential side effects. And further to that, promoting resistance. And so that's really not something that we want to do. So we really do have to change the way we think about and the way we use these antibiotics. So with that, I will stop blabbing. If there are any questions, we are happy to answer them.